Alright, so making a smear or a bacterial smear for looking at the bacteria under microscopes is pretty simple. What we are going to do is simply let things dry on here, right? So sometimes smears are called dry mounts because like with our cheek cell, we're going to add our bacteria, we're going to let it dry, and then we can color it, anything we want, right? So here's my, let's see, here's my labeled slide. All right, remember, always put samples on the opposite side. So just like before, we're going to flip things over, and we're going to have backwards labels, right? Now, we can put multiple smears on one slide. You can fit, if you're really good, you can put 5. I usually don't put any more than 4, but definitely 2 fits quite simple on here, right? So sometimes when you have multiple smears, it's really hard to label them all. So the very conventional thing to do is simply put A, B, C, or 1, 2, 3, or something like that, and then on some sort of sheet of paper, you have a code, right? So A is coli for me, B is Staph aureus, right? So then it allows me to go and check. But basically, it should be labeled in some way that when someone says, hey, what are you looking at under the microscope, you should know what you're looking at, right? But again, remember, sample side, opposite side of the labels. Now, when we add our smears, we actually want a little bit of liquid in there in order for everything to work proper. There we go, nice and sterile loop. We want a little bit of liquid in there. To be able to smear it properly, because the key to a smear is that we want to be able to spread it nice and thin, right? If it's too thick, it's very hard to color. It's hard to see what you want to see because everything's clumped up together, right? So we want to make sure that we can smear it nice and thin. Here I have my broth culture. This would be coli. Flame, right? Remember, always flame. And then to make a smear of a broth culture, it's simple because broths are liquid. So all I'm going to do is I'm going to take my broth and I'm going to smear it on here just like that. Okay, so I started in the circle first, but then eventually I start to smear it really thin, because the thinner your smear, the faster the next step, right, which is that air drying step. And you'll see sometimes that air drying step will take forever, right? So you want to make it nice and thin. I guess while I'm waiting for this thing to cool and sterilize, a key to putting multiple smears on one slide is that all smears should be about the same thickness, right? So in general, I like to put all type of cultures, or sorry, similar culture types onto each slide. So I put a whole bunch of broths on one slide and agar on another slide, just because it's easier to control similar types of cultures. Right? It's not a rule, it's just my preference, right? But today, because of demo purposes, I'm going to have one from broth. I wrote slant, but actually I picked a plate instead, so I'm going to grab one from the plate. Okay, so that's it. I'm just going to let that air dry. Alright, so now while that's air drying, I'm going to add my next culture, and this one is from an agar, and I'm going to grab it from a plate. Okay, now because plates are dry, I can't just simply grab the bacteria from the plate and stick it on there because it's too clunky, right? Too chunky, it's not going to spread well. So I have to add some liquid to that. So in order to do that, I have some water here. This is simply tap water, right? The water doesn't need to be sterile, but it does need to be clean, right? So some tap water in a beaker, just like that. I'm going to take a loop and then add my drop of water to that. Okay, now I'm going to add my bacteria to that. So now I want to sterilize because I don't want to get my culture contaminated, right? So while I let that cool, right? So the purpose of the water drop is to provide a little bit of moisture, so then I can spread my bacteria from the agar more effectively, right? Now again, when you grab inoculum from agar plates, don't grab a huge chunk. You don't want too much bacteria, right? So all I'm going to do is I'm going to touch it. Let's see, I'm just on the off camera, so I'm just going to just touch. That's it, and that should be way more than enough cells. I'm going to stick that, start in the circle, the water drop, and I'm going to spread slowly first just to make sure the stuff comes off into the drop, and then wide and big, right? Because I want to make sure that I spread it nice and thin. And the thinner you spread it, the faster the air drying time. And you'll find this one usually takes a little bit longer to air dry than the broth culture. But I thin big and wide and lots of spreading in order to let it air dry nice and fast. Okay, so that's done. I'm going to let that, I'm going to sterilize that. I'm done with the loop for my demo. Just to add that when you are making a smear off a slant, exact same thing because it's on agar, right? But instead of course, touching it down to the plate, you'd stick your loop down into the agar slant, and again, just touch. All you want is some bacteria, right? You don't want a huge amount. Okay, and then once that's air dry, and this one already, my broth culture that I put on here, my broth smear has already air dried. My slant is almost there, right? But because I spread that nice and thin, right? But that'll probably require another minute or so for that to fully air dry. Once it's fully air dried, we move on to our next step, heat fixing. After you heat fix it, then you can do any type of staining procedure with this, right? Whether it's a simple stain, whether it's a gram stain, or any other type of stain. Okay, now important that air drying is very important, right? Because remember, that little bit of moisture that's left on there can damage the sample when you heat fix it while it's still wet, right? And as I'm talking, I'm just down to a tiny little drop. I don't know if you guys can see that, so I'm just about done, right? So again, key, nice and thin. And that is how you make a dry mount or a smear from bacterial culture.